Hi to everybody. I am Silvia Vilasi from Italy, from National Research Council. I want to thank you for the invitation. I am very happy to be here, and now I share my screen. Also, on behalf of the many involved colleagues, I want to thank you for having supported our research by funding 2 years ago a grant for the project Identification of Compounds to Rescue MMACHC Functional Deficiency in CblC Disease. We are an heterogeneous group of biologists and physicists who work on proteins. And all we know that CblC is caused by modification in the gene codifying for a protein, which is MMACHC, which has a crucial role in metabolism of vitamin B12, a very complex molecule coordinated by a cobalt ion. That's why it's also named cobalamin, which is at the center of a chemical planar structure. It is bound to 4 nitrogen groups and also 2 other bonds to be satisfied, one in the region below this plane and the other one with an upper ligand, which can differ from one form to another of natural vitamin B12, in the sense that it can be an hydroxyl group, cyanide group, a methyl group giving rise to a methyl vitamin B12, or the oxyadenosyl group giving rise to adenosyl vitamin B12. These 2 forms are considered the active forms of vitamin B12 because they are involved in important reactions occurring in our cells, which are the transformation of acid methylmalonic and the transformation of homocysteine. So if these 2 reactions do not occur well, these 2 metabolites accumulate in our body, and this is the phenotypical manifestation of the disease. But who ensure that these 2 forms of vitamin B12 arrive there in the correct chemical form? There are a lot of proteins involved. Our focus is on the protein MMACHC, which has the role to transform vitamin B12 into its active forms. More specifically, the protein binds vitamin B12, and when this occurs, the bond between the cobalt and the lower ligand is broken. And then, with the help of some molecules like glutathione, the upper ligand is removed. But what is the structure of the protein? What does it mean, structure of a protein? When a protein was born, it is a linear chain of building blocks named amino acids. And then in time, this chain takes shape and assumes a 3-dimensional structure, which is strictly associated to its function. For example, in the case of MMACHC, this linear chain forms helices and sheets, which are arranged in the space to form a pocket to which perfectly accommodate vitamin B12. And another information, and that in the presence of glutathione, the ingredient necessary for the protein to function, it has been found that the proteins likes to travel as a couple, so to form dimers. The main goal of our research is to identify compounds able to correct the chemico-physical defects of MMACHC protein induced by pathological mutations found in CblC patients. So the first step is to try to understand what are these chemico-physical defects. What is this mutant structure? How do they react to the stress? Are they able to bind the vitamin B12 or to correctly transform it? Are they able to form dimers, which are probably the functional form of the proteins? Answer to this question is the starting point for imaging a therapeutic strategy for treating CBLC, and also the idea at the basis of our biophysical approach, which starts from obtaining the isolated forms of the proteins. We ask to bacteria to produce the proteins for us and to use physical techniques to study these proteins. Many of these techniques are based on phenomena of interaction, radiation, electromagnetic waves with the mother. These waves can be the light, for example. A phenomenon is the absorption of the light, responsible for the color of the objects, or the light scattering, which is diffusion. For example, when light is diffused by particles in the air, we can have the blue color of the sky, and so on. Obviously, our experiments start with the healthy proteins that has to be our reference. First, we try to understand if the protein MMACHC produced by us, for us by bacteria, had a correct structure. We used a technique named circular dichroism, which is also a sort of absorption of the light technique, and that gives information on the helices and sheet content of the proteins. We could verify that our proteins had a structure in good agreement with what was known from previous studies, and that this structure a little bit changed in the presence of vitamin B12, so indicative of the binding. Also, by looking at the protein with a highly focused and energetic X-ray beam, we could verify that this protein structure a little bit changed in the presence of vitamin B12. But we have a more direct and easy tool to verify by using biophysical techniques the binding of the protein to vitamin B12. Because, as I told you, when protein binds to the vitamin, the bond between cobalt and lower ligand is broken, and to this transition corresponds a change in the way the light is absorbed by vitamin B12. And we could verify that this change was present when to vitamin B12 we had the protein produced by the bacteria for us. And also that when to this mixed protein vitamin B12 we added glutathione necessary for the protein to function, the upper ligand of vitamin B12 was removed, and again changes the way in which the light is absorbed. So we conclude that the healthy protein produced by us works well. We have the tools to understand if it binds and transforms vitamin B12. Regarding mutations, we purified a point mutations resulting from a substitution of a building block of the protein with another one, and we purified a truncated protein resulting from a nonsense mutation in which we have a stop codon before the end of the protein, and so the protein is shorter than the normal one. We could verify that both these proteins did not function well as the wild type, as the healthy protein. And why do not they function well? What are the chemico-physical defects induced by these mutations? Regarding the first one, we could verify that the protein was able to bind the vitamin B12, but the bond was not so strong. We could verify this by using another physical technique that is fluorescence, by looking at fluorescence of the protein. And we also could verify that the protein, the mutant, was less stable than wild type. Because when we increase temperature, we can understand at what the temperature the structure begins destabilizing. And this temperature was lower for the mutant than for wild type. And also, we using light scattering, which gives us the information on the sides of proteins in solution, and by using native gel, which is a technique that separates species in solution on the basis of the size, of the charge, of the shape, we could conclude that the mutant was less able to form dimers than wild type. Regarding the other mutant, we could verify that the protein was able to bind vitamin B12, but with less affinity. The binding was also confirmed by the fact that the structure a little bit changed in the presence of vitamin B12 by looking at the technique I told you before, circular dichroism, and also by studying the movements of the protein during 100 nanoseconds, we could verify by using computational methods that this interaction mutant vitamin B12 is not stable. Since we found that both of these mutants were not stable, we tested the effect of some molecules named osmolytes able to stabilize proteins, not directly binding the proteins, but through the solvent, through the liquid in which the proteins are inserted in. We try to understand if one of these molecules was able to stabilize the proteins. And we tested betaine, already used in CblC patients treatment, and also TMAO, trialose, arginine, and we found that the only molecule able to stabilize both the proteins was betaine. And this also with another technique. We used the two techniques, fluorescence and circular dichroism, to verify that betaine was able to stabilize both the proteins and also to partially restore functionality of the mutant. So we tested as proof of principle that stabilization route could be a good route to restore protein functionality. And so next step was to identify molecules able to bind and stabilize proteins, so to bind the proteins. And the colleagues in Milano, our institute in Milano, identified a site between the interface of two proteins to be targeted with drug-like molecules from a virtual library made up of 1,000 safe molecules to be used with the human patients. And they identified 11 molecules that computationally were able to bind and stabilize the proteins. And we tested these molecules first in their ability to stabilize the protein, and we found that molecules 6, 7, 8, 9 were able to stabilize the proteins, and then in their ability to bind the proteins. And we used isothermal titration calorimetry, which by measuring the heat produced by the reaction protein molecules allows to understand the strength of the binding of these molecules with the proteins. This in collaboration with Adrián Velázquez Campoy in Zaragoza. And the two of these molecules overpassed both tests, and now we have to understand if they are able to restore functionality of the proteins. A good news is that a PhD student, Lisa Longo, by using another technique, I don't enter into details, found other 28 additional molecules by using a blind docking with no a priori hypothesis on the site to be targeted, and these molecules have to be tested. And then concerning nonsense mutations, because what I described so far mainly concerns missense mutations, so concerning mutations producing stop codons, often associated to nonsense mutated RNA decay, so the protein is not produced at all. We started a collaboration with researchers from our chemistry faculty, which work since many years on translational read-through inducing drugs, which are able to induce the production of the proteins also in the presence of these stop codons. mutations. And we are implementing cellular models of CblC to test these molecules on these CblC cells. Just to conclude, I want to say that a biophysical approach can be used to understand what are the chemico-physical defects caused by mutations found in CblC patients. So small molecules can be identified and can correct these defects, and that several nonsense mutations in CblC patients can be corrected also by using translational read-through using drugs. I finished. I will be happy to answer to your questions, and thank you for your attention.